Hello students of class 9th. I am going to start second chapter of chemistry, which is solutions. A solution is a homogeneous mixture of two or more components, and such type of solution which is made up of two components is called a binary solution. The two parts of a solution, binary solution is made of two components. And these two components are called as solute and solvent. The part of solution which is in small quantity or less quantity, it is known as solute, and the one which is in large quantity is known as solvent. For example, salt solution. Salt solution is made up of salt plus water. So salt is in less quantity, so it is known as solute, and water is in large quantity, so water is the solvent. Similarly, sugar solution. In sugar solution you have sugar plus water. So sugar is the solute and water will be the solvent. So what is the criteria for deciding what is solute and what is solvent? It is the quantity. If it is less in quantity, it will be solute. If it is more in quantity or large in quantity, it will be known as solvent. So a binary solution is made up of two components, solute and solvent. Now we will see that how a solution is classified as true solution, colloidal solution and suspension. So we will discuss that. We will be differentiating between the three types of solution. So on the basis of particle size, a solution is classified as true solution. Second is colloidal solution or simply colloids. Colloidal solution, or simply it can be called as colloids. And the third one is suspension. So we will be differentiating between true solutions, colloidal solution and suspension. So on the basis of particle size, those solutions in which the particle of solute is less than 1 nanometer, size is less than 1 nanometer. Now what is nanometer? Nanometer is 10 to the power minus 9 meter is known as nanometer. Whereas in colloidal solution, the particle size is in between 1 nanometer to 1000 nanometer. So you can see that the particle size of colloids are bigger than true solution, while in suspension it is more than 1000 nanometer. This was the first point. On the basis of particle size, we had differentiated. Now the second point we will see in terms of visibility. How does it appear in terms of appearance? A true solution is transparent. That means light can pass through it. Transparent. Whereas colloidal solution is translucent. Translucent, that it means that it allows light to pass through it partially. A small part of light is allowed to pass through it and other part is blocked. Whereas a suspension is opaque. That means it does not allow light to pass through it. Now the third point, a true solution is homogeneous in nature. Homogeneous means both the composition and the properties of the solution are uniform throughout the bulk. A colloidal solution, though it appears to be homogeneous, but is actually heterogeneous. Heterogeneous. Appears homogeneous, but actually is heterogeneous. And suspension is heterogeneous in nature. Heterogeneous means the properties and the composition is not uniformly distributed throughout the bulk. Bulk means from every part of the sample. Now we will see the next one. The next property is that true solutions can pass through filter paper. That means they cannot be separated by simple filtration. Similarly, colloidal solutions will also pass through filter paper, but they can be separated by using parchment membrane. Cannot pass through parchment membrane. Now what is a parchment membrane? I will tell you that. Parchment membrane can pass through filter paper. Now filter paper, the pore size is bigger, that is why colloidal solution can even pass through filter paper, but they cannot pass through parchment membrane, and suspensions, they cannot pass through even filter paper. That means they can be filtered. They can be separated by using simple method of filtration, because the suspension particles cannot pass through filter paper. But colloidal solution cannot be separated by filter paper because they can pass through filter paper, but they cannot pass through parchment membrane, so they can be separated by using a parchment membrane. So this part I am going to talk about what is a parchment membrane. A parchment membrane is a semi-permeable membrane. It does not allow the solute particles to pass through them, but can allow solvent particles to pass through them, because the pore size of parchment membrane is quite small, so solute particles cannot pass through them, but solvent particles can pass through them. So what is an example of parchment membrane? Animal skin is a example, or animal membrane is an example of parchment parchment membrane. So it is also known as semi-permeable membrane. So these are the points. You can take a screenshot to prepare the notes. I'm going to discuss the next points. 4 points we have already discussed. We will see the next points. So the next points are certain properties I will be telling you. These properties are new to you. You have not heard about them or you have not studied about them, but I will explain to you what it is. The fifth one is Tyndall effect. Colloidal particles show Tyndall effect, but true solutions do not show Tyndall effect. Now what is a Tyndall effect? That I will explain to you. First I will write down the point and then I will explain. Do not show Tyndall effect. Colloidal particles, they show Tyndall effect. Show Tyndall effect. Whereas suspensions, they show to certain extent. Show to certain extent. Now what does it mean? Certain extent means those suspensions in which the particle size is closer to colloidal solution, those suspensions will be showing Tyndall effect. Colloidal solutions show Tyndall effect, whereas true solutions, they do not show Tyndall effect. Now we are going to discuss what is Tyndall effect. Now Tyndall effect is, suppose you have taken a colloidal solution in a beaker like this. This is a beaker in which you have taken a colloidal solution. An example of colloidal solution is milk. Milk is a colloid. So when you take a colloidal solution in a beaker, and when you are carrying this experiment in a dark room, if you have got a source of light, suppose you have placed a source of light like this over here, a laser beam or an ordinary torch, the path of the light becomes visible. The path of the light becomes visible, and this property is known as Tyndall effect. A very common example or very common observation I will tell you about it. You must have seen that, especially in winters, when a ray of light enters the room, even the dust particles which are present are visible to us. Those dust particles are still present in air in front of you and in front of me. There are so many dust particles present in front of our eyes, but we are not able to see these dust particles. But sometimes when a ray of light enters the room, you can see that the entire path of the ray is illuminated, and very well we can see the dust particles present in the air. So dust in air forms a colloidal particle, and this shows Tyndall effect. That means the path of the light becomes illuminated, and this phenomenon is known as Tyndall effect. This is not possible. You will not observe it when you take a true solution. When you take a sugar solution or when you take a salt solution, you will not observe this effect. So true solutions, they do not show Tyndall effect. Colloidal solution show Tyndall effect, whereas suspension show Tyndall effect to certain extent. That means only those suspensions show the Tyndall effect whose particle size are closer to the colloidal range. So the next we will show, in terms of stability, we will be talking about stability. What do you mean by stability now? Stability, when we stand, when you leave it as some time, this is quite stable. Stable means that when you leave a sugar solution or a salt solution undisturbed, it will never happen that the salt and the sugar will get separated from each other. That means they will not, sedimentation will not take place. But in case of colloidal solution is also quite stable. If you will leave milk and water, it is quite stable. When you leave milk and water, even that will not separate. Milk and water will not separate into two layers. They will remain uniformly distributed. But suspension are unstable. Unstable means when you leave suspension undisturbed, then the suspension particles will get deposited at the bottom. Now what is the example of suspension? Soil and water mixture. Mud and water mixture forms a suspension. Chalk powder and water mixture forms a suspension. Sawdust, that is powder of wood, sawdust and water form a suspension. So when you leave a suspension undisturbed, then the two layers will separate out, and it shows that suspensions are unstable. If you take mud water, when you mix it for some time, mud will remain suspended in water, but after some time it will settle down when it's left undisturbed. So sedimentation will take place. Similarly, when you take chalk powder, when you simply stir the solution of chalk powder and water, then chalk powder will remain suspended in water for some time. But after being undisturbed, when it's left undisturbed, then chalk powder, whole of chalk powder will start floating on the surface of water. Similarly with the sawdust and water solution. So suspensions are unstable in nature. Upon left over undisturbed, they will separate out into two different layers. The next, so these are the properties that you have to study for differences between true solution, colloidal solution and suspension. Now I will talk about the components of colloidal solution. What is a colloidal solution made up of? Like a solution, I told you that a solution has got two parts, solute and solvent. Similarly, a colloidal solution has also got two parts. So the parts is, I'll rub this one. Colloidal solution has got two parts, colloids, and again they have got two parts. They are made up of dispersed phase and dispersion medium. Dispersion medium. They are similar to solute and solvent. They are similar to solute and solvent. So a colloidal solution has got two parts, dispersed phase and dispersion medium. Now I will talk about electrophoresis.